All right, so if you think you know eukaryotic cells because you've studied yeast or maybe human cells, get ready to have that foundation shaken a little bit. Today, we are diving into the absolutely fascinating and, frankly, kind of bizarre world of the kinetoplastid parasites. These organisms just don't play by the same rules of cell biology that we're used to. It's a whole other ballgame.
So, here is what we're going to cover. First, we'll figure out where these guys actually fit on the tree of life. Then we'll zoom in and look at what makes their cells so unique. After that, we'll get into two big-time human pathogens, Trypanosoma cruzi and Trypanosoma brucei. And we'll wrap it all up by putting them head-to-head and comparing them side by side. Sound good? Let's jump in.
Okay, first things first. Before we get into all the cool, weird details about their cells, we gotta ask a pretty basic question. Who are these things, and where do they come from evolutionarily? Let's find them on the tree of life.
Okay, let's start with a bit of a throwback to first-year bio, right? The three domains of life. You've got bacteria, archaea, and eukarya. Simple enough. Now here's the first big takeaway for us today. Our parasites, the kinetoplastids, are firmly in the eukarya domain. Yeah, they might be single-celled, but don't let that fool you. They've got a nucleus, they've got complex organelles. They're in our club.
So let's zoom way into that eukarya branch. And wow, look at this tree. It's just packed. You can see all the usual suspects, right? Plants, fungi, animals. We're all huddled together in this one supergroup called the opisthokonts. But just look at all this other diversity. We're going to ignore our own little branch for now and focus way over here on a different group, the one called the excavates.
And this right here is why I love biology. Things are always changing. This is a newer tree from 2020, and you can see how things get reorganized. Our own supergroup is now called Amorphia. You can see us, animals and fungi right there. But look, over here, the group is now called Discoba, which is where those excavates we just mentioned live. And right inside that group, boom, there they are, the kinetoplastids. This is the home of some notorious characters like Trypanosoma and Leishmania.
Okay, so we've placed them on the map. Now for the really fun part. Let's get out the microscopic magnifying glass and dive into the cell itself. Because I promise you, what we're about to see is a huge departure from the classic eukaryotic cell you learned about in your textbooks.
Just take a look at this thing. This is your basic kinetoplastid model. And yeah, you'll recognize some parts. There's a nucleus, a Golgi, a flagellum ripping around. But there is so much here that is just different. First off, they don't have a bunch of mitochondria. They have one single giant mitochondrion. And check this out. This little indentation here, the flagellar pocket, that's basically their mouth. It's where all the endo and exocytosis happens. And trust me, what's going on inside that nucleus, that's a whole other level of strange.
So why are they called kinetoplastids? Well, it's because of this incredible structure, the kinetoplast. Now, this isn't some separate organelle. It's actually a gigantic, super condensed network of mitochondrial DNA that lives inside that one big mitochondrium we just talked about. And get this, it's made of thousands upon thousands of little DNA circles all interlocked with each other. It's literally like a sheet of biological chainmail. You will not find this structure anywhere else in the tree of life. It is their signature feature.
Okay, if the chainmail DNA didn't convince you they're weird, their gene expression certainly will. You know how in most eukaryotes, RNA polymerase transcribes one gene, then stops, then finds another promoter? It's all very neat and tidy. Well, kinetoplastids just throw that rulebook out the window. They transcribe massive sections of their chromosomes all at once, in these long chains called polycistronic transcripts. It's way more like how bacteria do things. Then, they have this whole other complex process to chop up that long RNA strand into individual messages, and they have to stick on a special leader sequence through something called transplicing. The bottom line? Their entire system for making proteins is just fundamentally, radically different from ours.
So now we've got the basics of their biology down. Let's see how this plays out in the real world with a couple of heavy hitters. We're going to look at two parasites in the very same genus that have evolved completely different ways to make us sick. Let's start with Trypanosoma cruzi, the parasite that causes Chagas disease.
When you think T. cruzi, you need to think about the Americas. Its home turf is basically all of South and Central America. And because people move around, well, the parasite is moving around too, and its range is definitely getting bigger.
And there it is. That's the little guy right there. This is what you'd see in a blood smear. This particular stage is called a trypamastigote. And do you see that distinctive C shape it makes? That is the absolute classic textbook look of T. cruzi when it's hanging out in the blood.
Now, the disease this parasite causes, Chagas, is a really serious public health problem. It's tricky because it starts with an acute phase that can be super mild, maybe a little fever, or some swelling where you got bit. It's easy to miss. But the real threat, the insidious part, is the chronic phase. The parasite can just hide out in your body for years, even decades, without any symptoms. Then, for about a third of the people who are infected, it comes roaring back, causing these awful and often deadly heart and digestive system failures.
So how do you get this thing? Well, you have to meet its partner in crime, the vector. This is the Reduviid bug. It's got this creepy nickname, the kissing bug, because it likes to bite people on the face while they sleep. But, and this is super important, it's not the bite that infects you. It's actually way more complicated than that.
Okay, so here's the play-by-play. The kissing bug lands on you, takes a blood meal, and at the exact same time it defecates on your skin. The parasites are in the feces. So when the bite starts to itch and you instinctively scratch it, you're basically rubbing that infected material right into the bite wound. Or maybe you rub your eye or your mouth. Once it's inside, T. cruzi makes a beeline for your cells. This is a key point. It's an intracellular pathogen. It gets inside a host cell, makes a ton of copies of itself, and then bursts out to go find new cells to infect.
All right, let's switch gears and continents. We're leaving the Americas and heading to Africa to meet T. cruzi's cousin, a parasite with a totally different playbook. Meet Trypanosoma brucei, the agent behind African trypanosomiasis.
Right off the bat, everything's different. We're in Africa now, not the Americas. The vector isn't a kissing bug, it's the tsetse fly. And here is the single biggest difference in their strategy. T. brucei is exclusively extracellular. It never goes inside a host cell, not once. It just spends its entire life in you, swimming around in your bloodstream and your lymphatic fluid, completely out in the open.
This parasite causes the infamous sleeping sickness. It gets that name because in the late stages of the disease, it actually crosses the blood-brain barrier and totally messes up your nervous system and your sleep-wake cycle. But hold on a second. If it's always out in the open, swimming in the blood, how on earth does it survive the immune system? Well, it has this brilliant defense mechanism called antigenic variation. It's basically a master of disguise. It's covered in a protein coat, and it can just swap that coat out for a new one whenever the immune system starts to recognize it, constantly staying one step ahead of your antibodies.
Okay, so we've met our two main characters, two very closely related parasites, but they have taken completely different evolutionary paths to survival. Let's put them up on the board, side by side, and really lock in these differences.
I mean, look at this. The difference is just night and day. Lifestyle, T. cruzi hides inside your cells, T. brucei lives out in the open. Vector, kissing bug versus the tsetse fly. How you get it? One is from parasite-laden feces being rubbed in a wound, the other is a direct bite. Even the way they make you sick is different. Chagas in its chronic phase is thought to be an autoimmune problem, while sleeping sickness is a direct assault on your central nervous system. And maybe the most fascinating difference for us as scientists, how they evade immunity. We have a really good handle on T. brucei's antigenic variation, but how T. cruzi pulls it off while hiding inside cells, that's still a huge puzzle.
So this brings us to our final and maybe most important question. Think about it. We understand the molecular nuts and bolts of how T. brucei fools the immune system. It's a textbook example. But for its cousin, T. cruzi, we're still largely in the dark. So my question to you is this. How does that massive difference in our basic understanding affect how we even begin to think about designing new drugs or vaccines? How do you attack an enemy when you understand one strategy perfectly, but the other's is a complete black box?